how do we acquire knowledge in biology? We are going to acquire knowledge in biology by following very specific approaches. That approach is very scientifically based, right? Which means we will have to look at facts, not what we believe, not what we trust, not what we think, but what we see, what we can quantify. Science is factual, right? I want you to remember that. Science is factual. Science is measurable, right? It's evidence-based. If you don't have an evidence for something, don't try to give an explanation based on what you believe in, right? This is not science, okay? Now, science, it's a process, right? So to study biology, we're going to use the scientific method. That scientific method is going to be based on observation and question. So you start with an observation. That observation is going to cause you to ask a question. That question is going to trigger you to actually start looking for ways to explain the phenomenon you just observed. Now, a tentative explanation of the phenomenon, it's nothing but hypothesis, right? So you come up with a potential hypothesis. That potential hypothesis, you have to verify, is my hypothesis correct or is it incorrect? How do you go about that? Well, how you go about that is to design experiments. But before you can actually run your experiment, you have to come up with a good prediction, right? That prediction allows you to save money, to save energy, to save time. Now, that prediction is also what is giving you an idea of what could be the outcome of running that experiment, right? Okay? So, as you run your experiment, now you're going to gather results. You're going to analyze your result and draw conclusions. These conclusions can support your hypothesis or may not support your hypothesis. If the conclusions do not support your hypothesis, then you will have to design another hypothesis, then design some new experiment with obviously some predictions, and run some experiments again, gather result, draw conclusion, and see if now your conclusion are supporting your new hypothesis. And you're going to do that over and over until you can actually explain why what you think is happening is happening. All right. The scientific method is going to use very specific processes, right? There are different kinds of reasoning. You have what we call inductive reasoning and deductive reasoning. In the case of an inductive reasoning, you are going to work on a particular case, reach a particular conclusion. Then you're going to work on another case, reach the same conclusion. Then on another case, reach the same conclusion. As you do this over and over, you're going to come to the conclusion that, oh, wait a minute. All these cases are leading to the same conclusion. Therefore, actually, you can draw a general conclusion from all the small cases you had. That's inductive reasoning. That's the kind of reasoning that actually the guy called Fleming, who was a microbiologist, he was working on bacteria, and noticed that some organisms actually, when growing on his petri dishes, prevented the bacteria to grow. Okay, so this is when he came with the idea that, wait a minute, this organism that is preventing the bacteria I'm working with to grow, this organism actually has antimicrobial property. Therefore, this organism is producing antibiotics. And this is how penicillin was actually discovered and eventually later on isolated. Now, Fleming didn't isolate penicillin. Someone else did isolate penicillin. But let me close that parenthesis. Okay? So we have to be able to, okay, well, no, I was talking about inductive reasoning. And now let's talk about deductive reasoning. In a deductive reasoning, you are going to go from a general conclusion to a particular case. For instance, you know that mammals have hair, right? You see an organism that has hair. You're going to draw the conclusion that that organism has to be a mammal. Why? Because you know that the general assumption is that organisms with hair are mammals, right? So from general to specific, that's deductive reasoning. Okay? Now, I already tell you about the need for prediction and experimentation so that you can gather the results and draw conclusion. But when we do that experimentation, how do we approach things? Well, we have to make assumptions, right? We make assumptions, we design experiments, we have to have variables. We have to have, for instance, if I'm growing a plant and I want to understand the effect of water on plant growth, I'm going to measure the amount of water and I'm going to measure the impact of water on the amount of growth. Now, water that I control how much water I'm putting when maintaining my plant. That particular aspect of my experiment is going to be the independent variable, right? That's an independent variable. That independent variable, it's also called experimental variable. Now, the growth that I observe as I'm bringing water to my plant, that growth is going to represent what we call the dependent variable. And that dependent variable is the response to the independent variable. All right? Then, as we work with our experiments, how do we know that our experiment works or doesn't work, right? Well, we are going to know that our experiment works or doesn't work by comparing things, right? And what do we compare? We compare an experimental group or a test group to a control, right? By having a control, comparing it with a test group, we are going to see if our experiment works or it doesn't work, okay? Now, to acquire knowledge in biology, we are using a lot of models. These models allow us to extrapolate at some point to real life situation. For instance, the fruit fly, Drosophila melanogaster, is an organism that has been used for many, many years to understand the genetics in living organisms, including even human beings, right? How do we understand the passing down of genes from parent to offsprings? What are the patterns of inheritance? What are these patterns, right? Are genes that are passed down from offsprings to, from parent to offsprings, are they carried by some chromosomes that we call autosomes, or are they carried by sex chromosomes? If they are carried by sex chromosomes, what specific sex chromosomes? Is it the X chromosome? Is it the Y chromosome? Well, we know a lot of our genetics because of the study using Drosophila melanogaster, right? Then we have Sonorabilis elegans. Sonorabilis elegans is a unicellular, actually, is it a unicellular or multicellular? I don't remember. But Sonorabilis elegans is used to understand the physiology in more evolved organisms, including yourself. While Arabidopsis thaliana is a model of plant that is used to understand the genetics in plant. Mice are very much used to understand how we would respond to a vaccine, how we would respond to a drug, because mice happen to be very good model to study human diseases. And here we have a particular example of study, and in this study, actually the study focused on trying to understand the effect of the treatment of stomach ulcer in human by two new drugs. Okay, so we're looking at the two new drugs and we are asking which one is the most effective at treating stomach ulcer in human. And that stomach ulcer is caused by a bacterium called, what is the name of the bacterium again? It's called Helicobacter pylori. There we go, Helicobacter pylori. So we have groups of people. These groups of people, they all have stomach ulcer. But which drug is good at controlling, at treating the stomach ulcer? So now we are going to have people divided into groups. We're going to divide people because we have two antibiotics, antibiotics A and antibiotics B. We're going to have three groups. One group is a control group, right? That control group is going to receive a molecule that technically is not known to treat stomach ulcer. That would be the placebo, right? Then we have a second group that receive antibiotic A, and the third group that receive antibiotic B, right? The group that receives a placebo, it's called the control group. The two other groups are the experimental groups, right? So when we look at that then, and we actually do go put a probe in a person and see how the stomach ulcer is evolving with the treatment, right? What we notice is that at the end of the experiment, we see that only 10% of the control group who received the placebo actually got their stomach ulcer positively treated. However, in the test group with antibiotic A, we have 60% of the individual who experience an improvement in the treatment, actually cured, while test group 2, who actually took antibiotic B, has 80% of subject that got cured, which means that antibiotics B is the best one. Then, as we gather all this good information, and we want to tell the world that we actually found that of the two new drugs that have been developed, drug B is the best one, we have to publish it, right? But where do we publish it? We have to publish this work in peer-reviewed journals. Why peer-reviewed journals? Because the people who are actually assessing the quality of the work that was done, they have to be very specialized people. Remember, patients are actually going to be prescribed a drug. So we cannot just allow lay people to start saying, well, this drug is good. No, no, no. We have to see that this work was done with the best scientific practices, right? So here I'm going to show you some papers I published, right? So there is one paper I published in a journal that is called Expert Opinion, okay? That paper actually focused on antimicrobial drugs currently used and, the paper focused on antimicrobial drugs in phase 2 clinical trials and some drugs that were in preclinical trials for the treatment of tuberculosis. Remember, I told you that I used to work a lot on tuberculosis, right? And here is another paper I published. That paper, I published it in a scientific journal that is called Science of the Total Environment. Okay, this is another peer-reviewed scientific journal with impact factor, meaning that, you know, how much scientists are reading a paper, we are going to rate the paper, right? The higher the impact factor, the more people read that scientific journal, right? So you have some more here, neuroscientist, science, and then this other journal here. So we publish research discoveries in peer-reviewed scientific journals. But the problem with peer-reviewed scientific journals is that you have to be very highly skilled to understand what it talks about, right? It's not easy. That's why, in general, it takes another scientist to actually put the discoveries in the layman's terms so that they can understand what the discovery was about. All right, so in spite of all this discovery and all these advances, we still have a bunch of challenges. These challenges actually stem from, changes that happen in our environment, right? But also the new technologies that we are using. Well, when we are using technologies, we use it for our benefit, but these technologies do not go without causing some issues, right? What are some of these issues and how do we deal with that? This is going to be another research subject of study that needs to be done, right? So we acquire knowledge via science and we apply the knowledge by using technology, right? Because of, for instance, of what we see currently, climate change, we try to explain, okay, well, what causes climate change, right? Some of us are going to say, okay, well, you know what, the earth is just going through a cycle of high temperature and low temperature and high temperature and low temperature. Other people are going to think that no. You know what? You human, you are doing so much that you are causing global warming. For instance, what we see is that over time there is an increase in carbon dioxide amount in the atmosphere. Interestingly, as we observe that high amount of carbon dioxide that happened over the years, for instance here from 2007 to 2015, we have an increase of the particles of carbon dioxide in the atmosphere that are reaching above 400 parts per million, right? That increase happened to also match the increase in temperature. Therefore, we tend to draw a connection between the amount of carbon dioxide in the atmosphere and the increase in temperature. And we draw the conclusion that, well, you know what, humans, you are causing this global warming. You are causing this global increase in temperature. Okay? So that's basically by using science. So we have these challenges that we need to address. And finally, but not least, we have the challenge around biodiversity. As we look at biodiversity, we notice that biodiversity is increasing over time, increasing because of human impact on our environment. For instance, if you consider a new Costco that is being built, right? A new mall that is being built, we have to destroy a huge piece of land. We go into pour concrete and pour, asphalt. And this area that was at one time a forest inhabited by living organisms is not going to have this forest there anymore. So we're going to wipe down a number of organisms. That is a problem. We're going to cause habitat loss. And as a matter of fact, habitat loss account for the major threat on biodiversity. Let me repeat that. Habitat loss account for the most when it comes to threat on biodiversity. In fact, this small mammal that you see here, Molymus rubicola, which is native to Australia, has not been seen since 2009. Okay, which caused scientists to believe that, well, there is a possibility that this organism has gone extinct, right? Now, beyond that, we have emerging diseases that are a major issue these days, and re-emerging diseases. What are emerging diseases? Emerging diseases are these diseases that actually we didn't know of. They were not around. Now they are popping up. Okay? Re-emerging diseases are these diseases that we knew how to control, but now we don't know how to control. For instance, measles, right? Because of the success of vaccination, people in Europe and in the Americas don't understand that if we stop providing immunization to against measles, well, guess what? We are going to have measles around. And lo and behold, guess what? In the communities where you have a great resistance against immunization for measles, guess what? We see outbreaks of measles in these communities. All right? So this is an example of re-emerging disease. So some examples of emerging diseases we have: the avian flu, right? Avian flu, the strain H5N1, and the strain H7N9. These are two strains actually that were responsible for major outbreaks around the world. Swine flu, particularly the strain H1N1, it's a major problem with flu. SARS, the severe acute respiratory syndrome, you had a major outbreak that occurred in 2002, but that major outbreak got somehow confined, right? But eventually, in 2012, in the Middle East, there was a form of SARS that developed. But that form of SARS, we didn't call it SARS, we call it MERS, right? For Middle East Respiratory Syndrome. But the virus is exactly the same kind of virus than SARS. Now, closer to us right now, is SARS-CoV-2 or COVID-19. COVID-19, it's not just uh, an epidemic, it's a pandemic, right? We shut down the whole world because of this COVID-19, right? This speaks to emerging diseases. All right, what are the reasons for emerging diseases? There are many reasons, right? The many reasons it's that we have tremendously changed our lifestyle. We have tremendously affected the environment. We come in close contact with living organisms we didn't come in close contact with before. The type of farming we are doing these days, right? Now, mind that we have farmings for turkeys, right? We have farmings for bulls. We have farmings for all kind of organisms, including deers. As a matter of fact, where I used to live in Mineral City over 7 years ago, one of my neighbors had a farm of deers. All right, so that shows you how much we are coming close to some organisms we were not close with before, right? So we have an increased contact with animals. We have insects that can actually spread diseases to human, but mind that we are also human spreading diseases to animals, right? It's not only one way. It's not only human animals to human, it's also human to animals, right? Just keep that in mind, okay? The change in the human behavior, environmental changes, deforestation, the globalization of transport. Let's talk about globalization of transport. Right now, as I'm shooting this video, I am in Starr County, Ohio, right? Now, because of the transportation that are so effective today, in a matter of 7 hours, I can find myself in California if I take a plane. Assume that I was sick and I was having a nasty infection. I land in California, I will start spreading that disease around, okay? That's exactly what happened with the COVID-19 pandemic, right? From Asia, people traveled to America, to Europe, and to Africa to spread the disease there, which became a problem, right? For all of us, right? Okay? Re-emerging diseases because we are so successful at what we are doing. Now we say, okay, this disease doesn't exist. Okay, we say it doesn't exist because we've been using effectively vaccines and antimicrobials. Therefore, we have not seen the disease around. Because we have not seen the disease around, well, we think it doesn't exist, right? So that's not true, okay? So our own success, we become victim of our own success, right? So multi-drug resistant tuberculosis, I mentioned that. Extensively drug resistant tuberculosis, I also mentioned that as diseases that are now coming around. And finally, before we say goodbye, I want to remind you that please review the chapter, go at the end of the chapter in your book and go through the summary of the chapter or design yourself your own summary. Second, I want you to also look at the end of the chapter and test your knowledge, right? By reviewing with the questions that are at the end of the chapter, or design yourself your own questions, test your own knowledge, and finally draw some concept maps, right? Do it yourself with your own hands. Now, notice that I'm saying create a handwriting concept map. create a handwriting concept map. This is going to be tremendous. Don't type it. Write it with your hands, with pencil and paper, or with, an electronic pencil like the one I have, and a pad on which you can actually write. It makes a tremendous difference in terms of how you are learning. So thank you for your attention. Bye now.